Welcome to the Chemistry, Biology and Math Revision Hub. Today we are doing the Pearson Edexcel International A level, Biology Unit 2 for October 2023. Let's begin with the first question. Question 1. The diagram shows part of a cell at the start of metaphase 1 of meiosis. So this is the cell they're talking about. And they say which is organelle T. Organelle T is the centriole, because we see the spindle fibers are forming out of the centriole, so the answer should be A. And next they say explain why it is possible for chromatid A to be genetically different from chromatid B. A and B are sister chromatids that are originally identical, but because crossing over occurs in prophase 1, there is exchange of genetic material between the two non-sister chromatids, and that leads to chromatid B being different from chromatid A. So I said chromatid A and B come from the same chromosome. However, due to crossing over in prophase 1, it is possible for non-sister chromatids to exchange genetic information, resulting in A and B being genetically different. In part C, a student was asked to draw the 4 haploid cells produced by this cell in metaphase 1 of meiosis. The diagram shows the incomplete drawings produced by this student. Complete the drawings to show the 4 haploid cells produced. So if we go back here, we're going to have one like that, the next one like this, and then one like that, and the last one like that. So one has been drawn for us, which is this one here. The rest are the ones I drew here. Based on the information that we have, from here, this is what I have here. So this brings us to the end of question 1. Let's continue to question 2. In question 2, the photograph shows some pollen grains as seen using an electron microscope. So these are the pollen grains they're talking about. They put for us a scale bar here where the actual length is 20 micrometer. So they say calculate the magnification of this photograph. Give your answer to 2 significant figures. Using my ruler, I measured this to be 2.9 centimeters, and that's going to be my image size. Converting that to micrometer is 2.9 times 10 power 4 micrometer. In my magnification, which is image length divided by the actual length, or image size divided by the actual size, it gave me 2.9 times 10 power 4 divided by 20, which is 1450, and to 2 significant figures is 1500 as my answer. In part B, a pollen grain can produce a pollen tube that contains a generative nucleus. Plant fertilization results in the production of an embryo and endosperm tissue. The diagram shows the location of an embryo and endosperm tissue in a seed. So we can see the endosperm is here, and this is where the embryo is. They say describe how the generative nucleus results in the production of an embryo and endosperm tissue in a seed. As the pollen tube travels through the style, the generative nucleus is going to go through mitotic cell division to produce 2 male nuclei. One of the male nuclei is going to fertilize the egg cell, while the other is going to fuse with a 2-polar nuclei. So I said the generative nucleus divides by mitosis to produce 2 male nuclei. During double fertilization, 1 male nucleus fuses with the egg cell to produce the zygote. The zygote divides by mitosis to form the embryo, and the second male nucleus fuses with 2 polar nuclei to form the triploid endosperm. And in part C, flower color and pollen grain size are genetically determined in foxglove plants. The linkage of genes for flower color and pollen grain size has been investigated. State what is meant by the term linkage with reference to these 2 genes. If genes are linked, it means they're going to be found on the same chromosome because they're in close proximity with each other. So I say the loci of the gene for flower color and pollen grain size are in close proximity on the same chromosome. A statistical test can be used to analyze observed phenotype frequencies and the expected phenotype frequencies for the foxglove plants. Which of the following statistical tests will be used? This should be the chi-square test. It tells us if there is a significant difference between the observed and the expected results. So the answer here should be an A. So this brings us to the end of question 2. Let's continue to question 3. In question 3, some plant cell walls develop secondary thickening. Diagram 1 shows a cell without secondary thickening, and diagram 2 shows part of the cell walls of 2 adjacent cells with secondary thickening. So we have the first diagram and we have the second one. So they say give 3 differences between a cell wall with secondary thickening and a cell wall without. They want you to use the information in the diagrams to support your answer. Both have the middle lamella, but here we have more layers in comparison to that. Where there is secondary thickening, there is going to be lignin added and there is going to be low flexibility. So I said cells with secondary thickening have an extra layer compared to those without secondary thickening. And then they contain lignin, while those without secondary thickening do not. Due to secondary thickening, they are less flexible and impermeable to water. Then in part B, they say plant cell walls contain cellulose. How many of the following statements about cellulose molecules are correct? Cellulose contains alpha glucose molecules. This is wrong. Cellulose only contains beta glucose molecules. It contains 1,4 and 1,6 glycosidic bonds. That is also wrong. And then forms microfibules. This is correct. So the answer should be just one, which is B. And then next they say secondary thickening occurs in sclerenchyma fibers. Explain how secondary thickening contributes to the physical properties of sclerenchyma fibers. When secondary thickening occurs, lignin is added and this strengthens the fibers. So I said sclerenchyma fibers have greater strength because of lignification in the secondary cell wall layer. In part C, some cells in xylem vessels have secondary thickening. The photograph shows pits in the wall of part of a xylem vessel as seen using an electron microscope. We can see the cell wall and then the pits. They say describe the function of pits in the xylem vessels. Pits allow movement of substances from the xylem vessels into the neighborhood tissues. So I say they allow for the movement of water and mineral ions like nitrates or phosphates from the xylem vessels into the nearby cells. So this brings us to the end of question 3. Let's continue to question 4. Question 4. The photograph shows mangroves in the Caribbean. So these are the mangroves. They say a new type of bacteria was discovered living in these mangroves. Scientists call this new type of bacteria Thiaumargarita magnifica. One cell of this bacteria is 20 millimeters in length. The smallest known type of bacteria, Mycoplasma genitalium, is 300 nanometers in length. In part A, they say both of these are found in the domain bacteria. Give the names of the other 2 domains. One domain is eukarya and the other is archaea. Next, they say calculate how many times larger T. magnifica is than M. genitalium. Give your answer in standard form. I'm going to convert this and that into similar units. So if we have 20 millimeters, it's going to be 20 times 10 power 6 nanometers. So if we want to find how many times larger, we use this, which is 20 times 10 power 6 divided by the 300. After converting this to nanometers, this is going to be 300 nanometers. And then I got 6.77 times 10 power 4. So this is how many times larger it is. Then in part B, the cytoplasm of T. magnifica contains structures called peptins. The function of peptins is protein synthesis. Peptins contain circular DNA and ribosomes and are surrounded by a membrane. Explain how these 3 structures contribute to the function of peptins. The first one, they contain DNA. Of course, DNA contains the genes for formation of peptins. These genes are going to be transcribed into messenger RNA, which is going to be later translated into the protein. And then they contain ribosomes. This is where protein synthesis is going to occur, so the ribosomes are the site for the translation of the produced messenger RNA into a polypeptide chain, which is going to be the protein. And then they contain membranes. This means they are partially permeable to allow movement of substances. Some of these membranes are for compartmentalization or to compartmentalize so that specific conditions are provided for reactions to occur inside organelles. Also for formation of vesicles that enable substances to be transported within the cell or to the outside of the cell. In part C, some bacteria require oxygen in their surroundings, whereas other bacteria don't. The graph shows the growth rates of 2 types of bacteria, A and B, which were grown in different concentrations of oxygen. The bacteria were supplied with water and grown in their optimum temperatures. The key shows for bacteria type A and bacteria type B. So as the percentage of oxygen increases, the bacteria type B increases, while bacteria type A decreases. So bacteria type A is anaerobic, while bacteria type B is aerobic. So here they say explain why an optimum temperature and water are needed for bacteria growth. Because the optimum temperature provides the right kinetic energy for faster collisions between the enzyme and the substrate, so that the rates of reaction are faster. They require water for hydrolysis reactions during digestion, and also water is required as a solvent so that the substances that are digested dissolve into the water so that they can be absorbed by the bacterial cells. And next they say explain the results of these two types of bacteria shown in the graph. Like I've already explained from the graph, we saw that as oxygen concentration or oxygen percentage increases, the growth rate of B increases while the growth rate of A decreases. We also saw that at higher oxygen percentage, the growth rate of A becomes 0. This is because A is anaerobic. B continues to increase because it is aerobic. B also levels off because another factor other than oxygen concentration is limiting the rate of reaction. So this brings us to the end of question 4. Let's continue to question 5. In question 5, the lack of some inorganic ions in soil can affect the yield of fruit from crop plants. The photograph shows part of a vine plant growing in soil lacking magnesium ions. The vine plant shown has yellow leaves and reduced growth. We see the fruit on the vine plant and then the yellow parts of the leaf of the vine plant. They say which tissue is involved in the transport of magnesium ions in the plant. This is going to be the xylem tissues because these are the ones that transport water and mineral ions, and since magnesium is a mineral ion, it's going to be transported in the xylem. And next they say explain how a lack of magnesium ions could result in yellow leaves and reduced growth. Magnesium is required to make chlorophyll, and chlorophyll is used to trap sunlight during photosynthesis. So if there is less magnesium, there is going to be less chlorophyll made, there will be less photosynthesis taking place, there will be less glucose made, there will be less amino acids that can be made, there will be less proteins made, and there will be less growth or less structural proteins made for growth. Also, there will be less glucose for ATP production. So I say lack of magnesium ions leads to less chlorophyll formation. So lack of the green chlorophyll pigments makes the leaves yellow. Also, less chlorophyll results into less photosynthesis, less carbohydrate, less ATP, and less amino acids, therefore less proteins are going to be formed, less growth will occur. In part B, calcium ions are also needed for the growth of vine plants and fruit production. Chicken eggshells contain some calcium ions. Some farmers add crushed eggshells to the soil surrounding the vine plants. The effect of adding crushed eggshells to the soil surrounding the vine plants was investigated. The mean leaf area, mean shoot length, and mean chlorophyll content were calculated over 52 days. The graphs show the results of this investigation. On the vertical axis, we see mean leaf area of vine plant, and then on the horizontal axis, we see time in days. As time increases, there is going to be an increase in mean leaf area of the vine plant. There's going to be greater increase when there are eggshells that are crushed from about 24 days onwards. Before we have the same mean leaf area of the vine, that is before day 24. Down here we also have the mean shoot length on the vertical axis, and then on the horizontal axis we have time in days. There is going to be generally an increase in the mean shoot length for both, but there is greater increase when we have crushed eggshells than without. And here we have the mean chlorophyll content versus the time in days. Also we see as the time increases, again there is greater chlorophyll content among the experiments with eggshells that are crushed than without. So here they say discuss the benefits of adding crushed eggshells to the soil surrounding the vine plants. They say use the information in the question to support your answer. If we look at this experiment here, they have put error bars, and this shows us the experiments were repeated, but we can see there is overlap, and that makes the results not to be valid. So I said adding crushed eggs increases the mean leaf area length, mean shoot length, and mean chlorophyll content. The leaf area has the greatest increase between day 38 and day 45. Those without crushed eggshells had no change in the mean length area between day 38 to 39. For mean shoot length, the results had a slight increase until about day 25, After that, there was greater increase in those with crushed eggshells or crushed eggs. Data for the experiments with crushed eggshells for leaf area and chlorophyll content have error bars. There is overlap in the results showing they are not significantly different. Since crushed eggs contain calcium, it is used to make calcium pectate for the middle lamella. Greater leaf area provides more chlorophyll for sunlight absorption and more photosynthesis occurs. If there is more photosynthesis, this leads to more amino acid synthesis, more ATP through respiration, more mitosis, and more protein synthesis. So more structural proteins are made for increase in shoot length. More sugars can also be stored in the fruits. This also increases the size. So this brings us to the end of question 5. Let's continue to question 6. In question 6, a mimic octopus can change shape to look like other organisms. The photographs show a venomous sea snake, a mimic octopus looking like a venomous sea snake, and a mimic octopus looking like a flatfish. So we see the venomous sea snake, we see the mimic octopus looking like a venomous sea snake, and here the mimic octopus looking like a flatfish. The first part says this octopus is adapted to its niche. State what is meant by the term niche. A niche is a role over an organism in its habitat. The next they say the mimic octopus has the same banding pattern as the venomous sea snake. Name this type of adaptation. This is going to be anatomical because it's structure, but you could also think it's physiological because there could be reactions going on in the inside of the octopus that lead to these observable differences or observable changes. So it could also be physiological. Next they say suggest why the mimic octopus would change its appearance to look like a flatfish and a venomous sea snake. If it looks like a flatfish, it can camouflage from its predators or prey because camouflaging from prey allows it to get more food. Camouflaging like a venomous sea snake, many organisms are afraid of it, they will avoid it. In part B, the photograph shows a blue ringed octopus. The octopus produces a toxin called tetrodotoxin. The toxin affects neurons by blocking sodium channels. The pain-relieving properties of this toxin were investigated in a drug trial. The stage 2 of this drug trial compared the tetrodotoxin with a current pain-relieving drug in a group of cancer patients. Which of the following describes the purpose of the current pain-relieving drug in stage 2 of the drug trial? It's going to be a control because we are comparing. Describe the process that would have occurred before and after stage 2 in this tetrodotoxin drug trial. They could have done the pre-trials, then the stage 1, and after stage 2 they do a stage 3. So I said stage 1 would have been carried out by testing the drugs on a small number of healthy volunteers to determine if there is any side effects. And after stage 2, then stage 3 trial can be carried out on a large group of cancer patients. Stage 3 is going to be double-blind, and then statistical tests can be carried out to establish if the results are significant. In part C, the graph shows the duration of pain relief using both tetrodotoxin and the current pain-relieving drug in stage 2 of this drug trial. On the vertical axis, we have the number of patients, and on the horizontal axis, we have the duration of pain relief. We see the key, this is the tetrodotoxin, and this is the current pain-relieving drug. We can see that tetrodotoxin was more efficient in comparison to this one because the number of people with the pain relief were more overall. So they say comment on the results of this investigation. I said both drugs provide some pain relief. Tetrodotoxin was more effective because it showed greater duration of pain relief in more patients. The results have no error bars, so we can't comment on the significance or reliability of the results. And there is no information on the gender, age, cancer type, etc., so we can't appropriately comment on the reliability of the results. So this brings us to the end of question 6. Let's continue to question 7. In question 7, cells can divide by mitosis. The diagram shows a morula beginning to develop on the yolk of a fertilized fish egg cell. So we have the yolk and then we see the morula. They say which of the following describes a morula? A morula is going to be a solid ball of cells but this is going to be all totipotent, so the answer should be D. And next they say suggest one function of the yolk in the fertilized fish egg cell. The yolk is a source of nutrients, so I said it's a source of nutrients like glucose and lipids for respiration, as well as amino acids for protein synthesis. So this is going to allow for cell division, creating more cells, as well as growth of the embryo. In part B, as the fish embryo develops, different types of body cells and tissues are formed. One type of tissue that develops is skeletal muscle tissue. All the cells in the skeletal muscle tissue contain the same genes as the morula cells. Explain how these skeletal muscle cells can contain the same genes as the morula cells, but be different in structure and function. If this happens, it's because specialization has occurred. This involves epigenetic control, switching off genes, switching on genes, and so on. So I say the skeletal muscles are specialized cells. Although they contain the same genes as in the morula cells, some genes are not expressed in the muscle cells because they have been switched off. During cell differentiation, epigenetic control occurs where DNA methylation and histone modification occur. Some genes are switched off, so only active genes are expressed in the muscle cells. These expressed genes are going to be transcribed into messenger RNA, and then the messenger RNA is going to be translated into proteins at the ribosomes. So only proteins specific to the muscle cells are produced. This leads to differences in structure as well as function. In part C, the mitotic index of 4 types of cancer cell culture was investigated. The graph shows the results of this investigation. On the vertical axis, we have mitotic index as a percentage, and on the horizontal axis, we have the type of cancer cell culture. So here they say 2,000 OVCAR8 cells were studied. Calculate how many OVCAR8 cells were in interphase. So mitotic index is equal to cells in mitosis divided by total cells times 100. From this graph, the mitotic index is 5%. If X is the number of cells in mitosis, the mitotic index, which is 5, is equal to X divided by 2,000, which is the total cells times 100. And X is going to be 5 times 20, giving me 100 cells in mitosis. So the cells in interphase are going to be the total cells minus the cells in mitosis, which is 2,000 minus 100, giving us 1,900 cells. Next, they say which cancer cell culture has the highest proportion of cells undergoing nuclear division. The highest number of cells is the one with the highest mitotic index, and this is going to be HOC8. So the answer is going to be A. Explain why some people think that research into cancer treatments should not use embryonic stem cells. This is because it involves destroying the embryo, and there are some ethical issues around using embryonic stem cells because people think it's wrong. So this brings us to the end of question 7. Let's continue to question 8. In question 8, some organisms can produce gametes for sexual reproduction. The diagram shows an incomplete drawing of a human egg cell drawn by a student. So this is the egg cell, and they say state the letter which shows the zona pellucida. The zona pellucida is this one here. And then they say the student did not include the structure that contained enzymes involved in the hardening of the zona pellucida. Name these structures. The structures are called cortical granules. And in part B, the table shows some information about 2 birds. Both birds have a helical membrane around part of their sperm cell. We have the nuthatch, we have the red bunting, and then here with reproductive behavior, these female, they have one partner. These females mate with several partners or several male partners in a short period of time. So we can see the drawing of the head of the sperm. This has a shorter acrosome, greater nucleus or longer nucleus. Here we have a longer acrosome and a shorter nucleus. So they have different acrosome to nucleus ratio. Here we can see this area is wider. It has a wide helical membrane, where this one here is going to be narrow. They say how many of the following statements about sperm cells are correct. The midpiece of the sperm cell contains many mitochondria for anaerobic respiration. Mitochondria are not for anaerobic respiration, so that's wrong. The acrosome is a membrane-bound sac containing digestive enzymes. That's correct. The nucleus contains circular DNA. That's wrong. So the answer is going to be B. Only one is correct. Next, they say sperm cells with a larger acrosome to nucleus ratio have an increased swimming speed. They want you to calculate the acrosome to nucleus ratio of the red bunting sperm cell shown in the table. So acrosome to nucleus ratio using the red bunting cell, here you have to get this length using a ruler and then that length so that you can be able to calculate the ratio. I got 12.4 to 2.8 and my ratio was 4.4 to 1, which is what I wrote here. And next they say the effect of the width of the helical membrane on the mean swimming speed of sperm cells from different bird species was investigated. The graph shows the results of this investigation. On the vertical axis, we have the mean swimming speed of sperm cells. And then on the horizontal axis, we have increasing width of the helical membrane. We see as the width of the helical membrane increases, the mean swimming speed increases. And down here, they say the acrosome to nucleus ratio and the width of the helical membrane affect the swimming speed of the sperm cells. Explain how these adaptations are related to the reproductive behavior of these birds. Use the information in the table and the graph to support your answer. From the table, we can see the red bunting has a greater acrosome to nucleus ratio, and it also has a wide helical membrane compared to this one. These are all advantageous. From the graph, we see that increasing the width of the helical membrane increases the swimming speed. So here I said as width of the helical membrane increases, the swimming speed also increases. Red bunting females mate with multiple males within a short period of time. So the red bunting males need sperm with wider helical membranes to provide greater speed to compete with other males to fertilize the egg and pass on their genes to the offsprings. The red bunting, and again this should be red bunting male sperm, also has greater acrosome to nucleus ratio to have greater swimming speed to reach the secondary oocyte faster for acrosome reactions to occur. In part C, an individual organism that can produce both egg cells and sperm cells is said to be a hermaphrodite. Blue dragons are sea slugs that are hermaphrodites and can be found in the Indian Ocean. The photograph shows a blue dragon, which is this one here. They say each individual can produce both egg cells and sperm cells for sexual reproduction with another blue dragon. The sperm cells cannot fertilize their own egg cells, suggests reasons why it may be advantageous for blue dragons to be hermaphrodites. Because they are hermaphrodites, any blue dragon can mate or reproduce with any other blue dragon, and this is going to increase their chances of fertilization to pass on their genes to the offsprings. So I said each blue dragon can reproduce with another, and this increases the chances of fertilization, so more offsprings are produced. And since they carry out sexual reproduction, that sexual reproduction with others leads to greater variation in comparison to self-fertilization by one hermaphrodite blue dragon. In part D, the table shows some information about a population of 100 cats. So here we have the color of the cats, black, and white. Then we have the genotypes. We have the Hardy-Weinberg equation giving us the p squared, 2pq, and then the q squared. This is what was given. So they say complete the table to show how many cats would have the homozygous dominant genotype and how many cats would have a heterozygous genotype. Use the equation p squared plus 2pq plus q squared is equal to 1. So meaning q squared is going to be 16 over 100, which is 0.16. So rq is going to be 0.4. P is going to be 1 minus q, which is 1 minus 0.4, giving us 0.6. Q squared is going to be 0.6 squared, which is 0.36, but this is the same as 36 over 100. So that is going to be 36 out of 100. And the remainder is going to be 2pq, which is 2 times 0.6 times 0.4, which is 0.48, giving us 48 out of 100. So the answer here should be 36, and this is going to be 48. So this brings us to the end of question 8, as well as to the end of the whole paper. Thank you for being with us. Do not forget to subscribe. See you in the next video. Bye-bye.